Now let's take a look at transamination reactions followed by the urea cycle. These reactions, of course, will be primarily occurring in the extended fasting starvation state. The reason for this is because we want to use our primary energy sources of glucose and fatty acids first before we capitalize on using amino acids. Because amino acids are the building blocks of protein. And protein is broken down into amino acids due to the half-life of proteins. Now, in an extended fasting starvation state, we have a hormone called cortisol. And cortisol inhibits protein synthesis. By inhibiting protein synthesis, the natural half-life of a protein will cause degradation. The net result of this is to increase the availability of amino acids that can be used for energy. We typically do not want to use amino acids for energy simply because of using these building blocks for proteins to maintain our protein supply. Too much degradation of protein will compromise activity, physiological activity. So cells need to process the amino acids before they are able to use them for energy. And energy means oxidation. Now recall the structure of an amino acid. We have our amine, we have our central alpha carbon. Attached to this we have a carboxylic acid, and then we have the R chain that is useful for designating the function, structure, and function of the amino acid. Now recall the nitrogen here is a very electronegative atom, and so it is going to hang on to those electrons, and so those electrons are not going to be readily oxidized for energy. The rest of the amino acid is available for oxidative reactions. And by these reactions we'll be able to produce energy. So this means that the first step in processing an amino acid to be used for energy generation is to remove the nitrogen. All cells can perform this reaction, where we are going to take the amino acid of interest that we want to deaminate and convert this to the respective alpha keto acid. Now in doing so, we have to transfer the nitrogen. Loose nitrogen can generate free ammonia, which is dangerous. And so in order to do that, we need to transfer the nitrogen safely to a carrier. This carrier is going to be alpha ketoglutarate, and we are going to generate glutamate. This alpha keto acid that we generate here will be used for oxidation and generate energy. Now the glutamate can either be secreted into the blood, or it can be further processed to glutamine. And this is by glutamine synthase to process more ammonia. This reaction essentially is going to take the amino group of an amino acid and place it onto the R chain, the side chain of glutamate. So now the alpha ketoglutarate has received two nitrogens, one from the donation from an amino acid, the second one from glutamine synthase to generate glutamine. This reaction here, the primary reaction of swapping an amino acid nitrogen onto alpha ketoglutarate, this reaction is called a transamination. Transamination reactions can occur for all amino acids, and this is the way we process and get rid of the nitrogen in the first step. Now once the glutamate or glutamine enters the bloodstream, these are taken up by the liver. For skeletal muscle has a slightly different pattern of disposing of nitrogen because of its high energy demands. So skeletal muscle is going to be more likely than any other cell to oxidize amino acids into energy. What skeletal muscle will do is it will take pyruvate. And let me draw the structure out here so you can see the transamination reaction. Here's pyruvate. And we will undergo a transamination reaction to generate the amino acid alanine. Now notice what happened here. We took an amine from another amino acid, generated its respective alpha keto acid, and we placed that amine onto what is essentially the alpha carbon of the amino acid to generate alanine. If this was the reverse reaction, we would remove this nitrogen and return the carbonyl on the alpha carbon. This is why the pyruvate is considered the alpha keto acid of alanine, because of the alpha carbon. In this acid form here, we have a ketone, alpha keto acid. Now, skeletal muscle is able to process amines by putting them onto pyruvate and generating alanine, and then this is secreted into the blood. This will eventually become what's called the alanine glucose cycle. Recall this cycle from gluconeogenesis, because once the alanine is in the blood, the liver takes up the alanine, undergoes a transamination reaction to pyruvate by the enzyme ALT, alanine transaminase. This is one of our enzymes we test for liver function. Because there's so much alanine coming into the liver from skeletal muscle processing, we have a large expression levels of ALT, and that's why it's a good marker for liver damage. Once the liver converts the alanine into pyruvate, the pyruvate then enters gluconeogenesis. Now let's go back to the other products of transamination reactions, glutamate and the glutamine synthase reaction of glutamine. These, of course, will be taken up by the liver. And we need to talk about this in terms of mitochondrial matrix and the cytosol. So this, of course, is the inner mitochondrial membrane. And these two compartments of the hepatocyte is going to be processing the nitrogen in order to get rid of it as urea. So the glutamate, or glutamine, will enter and be transported into the mitochondrial matrix. Glutamine is processed to glutamate by the enzyme glutaminase. This reaction generates an ammonia. Glutamate in the mitochondrial matrix of the liver will be processed to alpha-ketoglutarate by the enzyme glutamate dehydrogenase. Being a dehydrogenase, this is also going to generate some energy for the liver. Importantly, this is also going to generate a free ammonia. The alpha-ketoglutarate, of course, can enter the TCA cycle, or it can be transported into the cytoplasm. We'll take a look at the cytoplasmic form in a minute. Now, free ammonia is rapidly diffusible, and so the free ammonia, once it's generated by the glutaminase or glutamate dehydrogenase, needs to be trapped very quickly. And this is trapped by combining the nitrogen with bicarbonate. Recall that the bicarbonate is being generated from TCA cycle, the CO2 being generated, dissolved in solution. The bicarb will be combined with the ammonia and form carbamoyl phosphate. The enzyme that does this is carbamoyl phosphate synthase. This is a very rapid enzyme, and as soon as the free ammonia is generated, it's trapped with the bicarb ion. Now, still in the matrix of the mitochondria, the carbamoyl phosphate is going to be combined with ornithine. Ornithine is an amino acid. It's not one of our 20 amino acids that's used to be making protein, but it is an amino acid here. This enzyme is ornithine transcarbamylase. Ornithine transcarbamylase is one of the enzymes that's targeted in mutation that generates urea cycle defects. A mutation in this enzyme, as you can imagine, is going to back up the synthesis of carbamoyl phosphate, inhibiting this, leading to the generation of free ammonia and hyperammonemia. This reaction will produce citrulline. Now, at this point, the citrulline is able to exit the mitochondria and enter the cytoplasm. This is done by an antiporter, and this is the citrulline ornithine antiporter. Now, once the citrulline is in the cytoplasm, it can now undergo the next reaction to generate argininosuccinate. To generate this product, we are going to be adding aspartate. Aspartate will be donation of the number 2 nitrogen on our final product of urea. Recall the structure of urea has 2 nitrogens connected by a carbonyl. So the first nitrogen that is donated is going to be donated from carbamoyl phosphate, and that is going to be in the form of free ammonia. So this is carbon number 1. The second nitrogen. The second nitrogen that will be donated will be coming from aspartate. This enzyme is called argininosuccinate synthase. Now the next step of this reaction is going to be mediated by argininosuccinate lyase. And by this reaction, we will be creating the amino acid arginine. This enzyme, argininosuccinate lyase, will be releasing a fumarate intermediate. We will process this fumarate in a second. The arginine will then be processed by arginase to generate ornithine, releasing urea from this step, our final product. Once the ornithine is formed, now the ornithine can be used in the co-transport, in the antiport transport of citrulline. So for every citrulline that comes out, one ornithine goes in to balance our chemistry and our cycle to continue. Now important is the regeneration of aspartate, nitrogen number 2. This reaction in the mitochondria matrix will continue as long as we have glutamine and glutamate coming in to the liver cell from extrahepatic tissues, generating free ammonia, generating carbamoyl phosphate. So the flow of nitrogen coming through here is consistent from extrahepatic tissue. Now what about the second nitrogen? Well, let's take a look at fumarate. Fumarate, we have an enzyme, recall, fumarase, that is going to be generating malate. Recall this is just an addition of water. Fumarate has a double bond to form malate. We also have malate dehydrogenase in the cytoplasm, which will be generating some NADH from the malate, and we are going to form oxaloacetate. The final step is to recall that oxaloacetate is the alpha keto acid of aspartate. So the oxaloacetate will undergo a transamination reaction with the enzyme AST for aspartate transaminase. So this is our second liver marker. The first one was ALT. The second one is AST. Now we have to consider we are donating a nitrogen here. This is a nitrogen acceptor. So where is the donor? The donor will be glutamate, 2-alpha keto glutarate. So this is a transamination reaction that capitalizes on glutamate being in the cytosol. So part of the glutamate from extrahepatic sources will be entering the mitochondria to generate free ammonia to supply the number 1 nitrogen. Other sources of the other place for glutamate is in the cytosol so that we can restore the number 2 nitrogen donor, which is aspartate. Now as far as regulation of the urea cycle, regulation is strictly by substrate availability. In other words, the urea cycle is in the liver, in the hepatocyte, ready to go. The enzymes are available, the reactions are ready, and as soon as substrate becomes available. So under a mild fast, the urea cycle will be moving slowly. As we extend our fast into a more severe fast, when we are going to capitalize on amino acids for energy, now the urea cycle will move much faster. Any defects in any of the enzymes in the urea cycle will, of course, build up ammonia. Buildup of ammonia is called hyperammonemia. The other way to cause hyperammonemia is to saturate the capacity of the urea cycle. So if somebody diets on strictly amino acids with no glucose or lipid sources for fuel, these amino acids will be processed for energy. For example, bodybuilders that might go on a strict protein or amino acid diet. It is possible to saturate the urea cycle with the processing of these amines while using amino acids for energy. This will also lead to hyperammonemia.